Hello, my dear students. How are you? I hope that everything is going well in your lives and you are in good health. I hope also for you a happy new year. Today in this session, we're going to read together Activity 1, Examples of Animal Cells. We talked before the vacation about types of cells. We said that the cell can be a plant cell or an animal cell. We already studied everything about plant cells and we took examples about them. The animal cells, they can be taken from a unicellular organism or multicellular organism. The unicellular organism is the organism that is made of one single cell, the paramecium, while the multicellular organism is the organism that's made of uncountable number of cells, like us, the people. In order to make preparation for paramecium, page 42, we need to make these steps. Now, you don't need to memorize the steps, just read them. We have to bring water from nature, like rainwater, store them in a jar with some dead leaves, close them with a cloth and rubber band, then store them in a closed, dark closet. Why? In order for the paramecium to multiply. Maybe in the water that you got from nature, there is only one or two paramecia. Now, they are microscopic, so you can't make sure that you are going to be able to observe the paramecia under the microscope. That's why we need to prepare large number of paramecia. That's why we put them in a closed, dark closet, so the paramecia can multiply, يعني they increase in number. Then, after 1 week, we take a drop from this water, we put it in a slide. The jar will contain uncountable number of paramecia after 1 week. So when we take one drop, this one drop will contain some paramecia. The paramecium need to be colored, so we use iodine water, for example, methyl green, and neutral red. These are other examples about other dyes that we use in the lab. Then, of course, we cover it with the cover slip. Now, the is ready to be observed under the microscope. We are going to observe this animal. It is a unicellular organism made of only one cell. This cell can breathe, can eat, can move. It has many parts. It has cilia, vibratory cilia. They cover the whole cytoplasmic membrane. They look like hair and they allow the paramecium to move. The cilia allow the paramecium to move. Then we have the cytoplasmic membrane. It encloses, it surrounds the cytoplasm. But the cytoplasm is granular. Granular cytoplasm means that the cytoplasm is not clear. There are like tiny particles in it, as if there is sand in it. We have two nuclei, one big nucleus and one small nucleus. And we have many vacuoles. The one that look like a flower, up and down, they are called pulsatile vacuole. Pulsatile vacuole. While the others that look like circles or they have round shape here, here, and here, and here, they are called digestive vacuole. I'm going to repeat the names again. We have cilia, say them with me. Cytoplasmic membrane, pulsatile vacuole, digestive vacuole, granular cytoplasm, a big nucleus, and a small nucleus. The paramecium has an elongated shape. It looks like a slipper actually. Now, I asked you before the vacation to draw this drawing on your copybook. I hope that you drew it in a good way and in a neat way. Now here this figure, it shows a paramecium that is observed through a highly powered magnification, يعني by a microscope that's very advanced and it can enlarge in a very good way. This is everything you need to know about the paramecium. You have to know how to draw it and to label it. Now move to the preparation of a cell that is taken from a multicellular organism. This cell is taken from the human body itself. It's very easy to prepare this preparation. We simply bring a toothpick, we rub it inside of our mouth from the inside of the cheek. Then we put the scratch or the things that we got on the toothpick on a slide. We should color it. So what should we do in order to color the cells that we got? We color them by iodine water. Then we cover the preparation by cover slip. Easy steps, simple steps, you need to memorize them. Inshallah, when we go back to class, we are going to make these steps in the class and you will observe the cheek cell and the paramecium. Now, the preparation is ready to be observed under the microscope. We are going to observe the inner wall of the cheek, the cells of the inner wall of the cheek. This is what we are going to observe. Flat cells. Flat cells means that they look like the bread loaf. Okay? It's like a disc shape. They have this shape, very simple parts, only cytoplasmic membrane from outside, granular cytoplasm. Also, the cytoplasm has granules in it. It's not clear. It seems that as if we put sand in it. And we have, of course, the nucleus. There's only 3 parts in the cheek cell. You need, Kaman, also I asked you before the vacation to draw and label the cheek cell on your copybook. I hope that you drew it in a good way. This is everything about the paramecium and the cheek cell. So, the paramecium has many parts, while the cheek cell has only 3 parts. Try to memorize them and to draw them. This is everything for today. And inshallah, I'm going to continue next session. Inshallah, we hope that next sessions are going to be in the class. Have a nice day, everyone.